this one, and this one. Okay.
I want to walk you through both of these because both of these have some tough issues that might not appear obvious because man, that they not appear obvious to me. So this is the beginning of an article that looks at tissue by tissue what is happening in a patient who is insulin resistant. Okay. I found the part that I bracketed to be kind of interesting, so definitely read that. Okay, and I took notes on it. I looked at this kind of flow of events that lead to insulin resistance. And then, right after that brackets, or it says at the molecular level, okay, part of the problem, or a huge part of the problem, is the serine phosphorylation of the insulin receptor substrate. That's where we're going, okay? So that's what you got to keep your eye out for in the next couple of days. All right, then the next page, this is the one that kind of blew my mind here. It took me forever, and I did not print it up in color, even though it said that some things are shown in red and some things are shown in green. Well, no, they weren't. So they must have changed their mind as they were publishing the data. Okay, so take a look at take a look at muscles. Okay, I'm just going to interpret how you read this chart. It says there's a box with an R in it and a box with an R in it. Increased glucose uptake. Increased glycogen. Okay, so this is how you interpret it. This is what insulin is supposed to do. So the event afterwards. Okay. And the R or the S tells you, does this particular response become resistant to the presence of insulin? Okay, so you'll notice that a lot of those normal insulin responses become resistant in people who are developing insulin resistance. Okay, but some things remain sensitive. So even though a lot of activities become insulin resistant, some things never become insulin resistant. So the presence of insulin maintains those particular activities. Okay? So R means these activities become insulin resistant. And if you see an S, those activities remain sensitive to the presence of insulin. Okay? So that's how you read it. So take a look at that. One of the things I want you to look for on the first page as you're reading through the physiological effects or events that lead to insulin resistance, notice how many of those are causes of ER stress. So later, in a few minutes, I'll list the causes of ER stress. And then I want you to take a look at how many of those things that you find in people who are developing insulin resistance are actually ER stressors. Okay, so that's that handout. If you want to get the whole article, you can get the whole article. It's free online. Okay, the second handout is some data interpretation from an article that I found. And I typed out the reference at the bottom, and then I thought, No, I don't want you guys looking at the reference, because then you're gonna let the authors do the work for you. So, it's from 2013. So actually rather recent. Okay? So one of the causes that I'll list about ER stress is the loss of calcium homeostasis. Too much calcium is entering the cytoplasm. That's one kind of a stress. Another kind of a stress is too much calcium is leaving the endoplasmic reticulum. Okay, so the ER is the place where calcium is stored. Okay, and the homeostasis of calcium is a balance between calcium going into the ER and calcium leaving the ER. Okay, so that balance has to be maintained almost all of the time. If not, that is a stressor for the cell, and that elicits the unfolded protein response. Okay? This is done by Circa. Okay, this is we covered this one, it's the calcium ATPase. Okay, we identified the organelle as sarcoplasmic reticulum, but it's the same thing as endoplasmic reticulum. And on the other side, there's a calcium leak channel. Okay? But nobody has yet identified what is that calcium leak channel, because calcium is not going to cross the membrane no matter what. It does not diffuse at all. Unlike glucose, it is not crossing the cell membrane. So there has to be a calcium leak channel. Well, the authors of this mystery paper thought, oh, I bet it's the translocon itself. Okay, so that's the big question mark. Okay, so in this paper, that was the question they asked. Could it be the translocon that is the calcium leak channel? Okay. So they did a variety, they treated cells with a variety of different inhibitors, and they looked at the amount of BIP being produced. Okay? I'm going to try to stay middle. Unfolded protein response can be measured by looking at the amount of BIP. If you activate the unfolded protein response, you see a buildup of BIP. Okay, and we'll see why in a minute. And we'll also see how, too. Okay, so wherever you see a lot of BIP, and actually all you need to do is focus on figure B. Figure A shows a Western, and it was these Western results that led to this particular figure B, okay? But just focus on B. The height of the column really is the amount of BIP present in the cell. So a lot of BIP present in the cell means the unfolded protein response has been activated. The cell is under ER stress. A low bar means that, well, there's just a typical amount of BIP present in the cell. The cell did not activate ER stress. Okay? And I told you also, what are these, why did they use these inhibitors? What are they accomplishing by the addition of these inhibitors? Okay? So, the question that you need to answer, just looking at figure B, okay, is, could the translocon be the calcium leak channel? And how do you know? How do the results show that? So if you say yes, how do the results show that the calcium the translocation is a calcium leak channel? If the answer is no, how do the results show that it is not the calcium leak channel? Okay, so this, by the way, is the format that homework number 2 that I'll give out to you in a couple of days, probably Friday. That's the format of the homework that you're going to get next. Is you're going to see some data. Everything from the paper that I could remove from the paper has been removed, so you cannot use the author's conclusions. You have to come to your own conclusions. Okay? And that's exactly, I don't know, at some point when you've read a whole bunch of papers, you just don't even bother reading the text. You look at the figures. Because you don't need to read the text anymore. So I just wanted you to do the thinking, because if the text is there and you can see the paper, you spend way too much time trying to figure out what the authors are saying. Right? And it's all up here. You can do it. Okay? It's problem solving. Okay? So those are the 2 things I want you to do in lieu of me not being here tomorrow. I think that was fair. I didn't cheat you guys out on an educational opportunity and program.
Do you want us to write something about this and turn it
You
in,
know,
or just?
oh God, I'm kind of up to my eyeballs in grading right now. So no, but we'll talk about it in class. And, kind of like the experiment we did on the last unit, something might appear on the test. Okay? So we will talk about it briefly. But you could practice. So when we talk about the answer, we'll talk about what constitutes a good answer to the question, how do you know, and what constitutes an inadequate answer to the question, how do you know. So that's kind of how I will use this assignment, is to make sure you understand what things need to go into the question, to the answer to, so how do you know. Anybody look at who won the Nobel, or who won the Nobel Prize in medicine and what the topic was?
Was it I forget his name, was it the Japanese cell
I don't even know the name, but what was his research topic? He was working on a process in the cell.
Recycling?
Huh?
Was it something about recycling, like biological recycling? Or?
Sort of. It's autophagy, where the cell eats parts of its components. So a worn out mitochondria is chewed up by autophagy. Or worn out proteins can be engulfed in an autophagosome, so a membrane forms around these things that need to be degraded. And you can get rid of protein, organelles, you can get rid of proteins that way. So we notice that the proteasome is one way of getting rid of proteins. Autophagy is another way of getting rid of proteins that are no longer needed. Okay. Autophagy is sort of a bulk process, although the contents are usually selected in the autophagosome. So I tried my best over the last 10 years to pretend autophagy didn't exist, and I just can't do it anymore. We had a whole biochem seminar, a whole semester on autophagy once. I couldn't ignore autophagy, so we're going to spend some time at the end of the semester looking at autophagy. So we'll probably have to go read the Nobel Prize lecture that they did about autophagy. That might be a good way of looking at it. Okay, so the unfolded protein response. Yes. This is, once we go through all of the events of what is the unfolded protein response, then you're going to understand why we have cheat sheets. Okay, so hang on to your hat. Oh, you kind of need a poster size. Okay, so far, I'm not going through any of it. We're just going to go through, I'm going to talk about what things cause the unfolded protein response and what are the overall results of the unfolded protein response. And then we'll get into the nitty-gritty detail. Okay. Okay, so the first thing I want to look at, and my list is long, are what kind of stressors cause ER stress. Okay, so this is a huge list. Inflammatory signals cause ER stress. And sometimes in fact, most of the time, I don't understand the relationship between the factor and the initiation of ER stress. I understand the unfolded protein response. I don't understand how inflammatory signals cause ER stress, except that apparently they do. Okay? A lot of viruses cause ER stress, and that's understandable. In producing new viral particles, a lot of those viral proteins have to be made up by the secretory pathway. So you get an influx of huge amounts of viral proteins in the ER. That one I can get. Genetic mutations. Okay, so like, for example, cystic fibrosis, where the cystic fibrosis transmembrane regulator, that chloride channel gets stuck in the ER because it doesn't fold properly. In emphysema, I think the enzyme elastase gets stuck and bound to calnexin or calreticulin. It just cannot fold, it cannot fold, and it cannot fold. Okay, so you have a mutant protein that has to go through the secretory pathway, and it remains unfolded. That causes ER stress. Okay, hypoxia. What's hypoxia? Hypo means not enough of, and oxia, not enough oxygen. Okay, so when the cells are not exposed to enough oxygen, that can cause ER stress. Ooh. Be a longer list than this. I think we could, do I want to say saturated fatty acids? Yeah, especially saturated. Okay, so when there's an influx into the cells of free fatty acids, particularly saturated fatty acids, that can cause ER stress. Okay, we already talked about normal development. Or differentiation. Pancreatic cells, both the insulin producing cells and the cells that produce digestive enzymes have huge amounts of endoplasmic reticulum. And that's because at one point in their development or differentiation, they went through the unfolded protein response. They responded to that by making a whole lot more ER than they used to have. Okay, so that's how a cell gets a whole bunch of endoplasmic reticulum. Okay, calcium imbalances. That's what you're looking at. Okay, it could be a buildup of calcium in the cytoplasm. It could be the loss of calcium from the ER. And this particular paper, if you read the text, they talk about the imbalance as a loss of calcium from the endoplasmic reticulum. But you don't have that text. Oxidized fatty acids. I'll just say fats because it doesn't necessarily have to be just fatty acids. And there's probably more, but those are probably the ones that have been studied the most. So take a look at that article on insulin resistance. How many of these factors are present in a patient who's developing insulin resistance? Okay. So here's what the unfolded protein response includes. If the ER is already filled with proteins that it can't manage, it doesn't make sense to add more proteins. Okay, so there's a global decrease in protein synthesis, although some messenger RNAs are allowed to escape that particular block. Okay, and as a matter of fact, a more recent event in the unfolded protein response is that some specific messenger RNAs are degraded to keep protein from entering the ER. So here's a group of proteins that have to escape that inhibition of protein synthesis. You have to have more protein folding chaperones. For example, BIP. Okay, so that's why the authors of the experiment that you're looking at used BIP as a measurement of how much unfolded protein response is going on here. Okay, an increase in the synthesis of lipids. Okay, predominantly phospholipids. Oops. Okay, so these two guys together, more phospholipid synthesis, more more protein folding chaperones, this results in more endoplasmic reticulum. Okay? So those are the events of let's just get more endoplasmic reticulum, and we can now have more space to accommodate all of the proteins that are going through the endoplasmic reticulum. So this is a good adaptation strategy. And then to help relieve some of the stress in the ER, we need more ERAD proteins. Okay, proteins involved in degradation. Okay, so most of the time, cells will adapt. They'll build more ER. Okay, they stop loading the ER with proteins that need to go through the secretory pathway. They build more ER, they get rid of a lot of the proteins that were in the ER. Okay? If a cell doesn't adapt, the cell will commit apoptosis. Okay? So when we cover apoptosis in the last unit, we'll bring back ER stress in the unfolded protein response to look at how does ER stress cause apoptosis. But for now, we're just going to be looking at this. So the cells are going to go through the unfolded protein response, and eventually they'll adapt. They're not gonna die. Okay, so here's where things heat up. Your book sort of covers extremely vaguely, and I think inadequately, the unfolded protein response. You might want to be looking at the figures in that first article that I put online. Okay, it's called article number 1, and it's I think they're getting to be an old article, but it's just great. It's great. Okay, so there has to be some proteins in the ER that recognize, well, something's wrong here. Okay? So there are 3 proteins that really play the role of ER stress recognizers. and they are PERC, which we probably won't cover today. So that meant I had to, that's why the one assignment I wanted to give you, I couldn't give you because it requires that we covered PERC. Okay, IRE. Which is the first one we're going to go through, and ATF 6. Okay, these are the things that are sensing ER stress, and they become activated in response to ER stress. Okay, it's amazing. You know, so each one. Turns out that each one is going to activate a transcription factor that will turn on the synthesis of these genes. Okay. But they're going to be doing a whole bunch of other things too. So that's what we have to cover. So we're going to cover IRE first. It's the hairiest of the three ER stress sensors because it's sort of like those kinds of those cleaners that don't clean anything, like Mr. Clean or something. It's the multi-purpose protein. It does everything. Okay, so you can, when you're drawing pictures, you can make IRE look like Mr. Clean. Okay, I'm going to try not to get too close to the edge. Of the proteins, PERC, IRE, and ATF6 are transmembrane proteins of the ER. Okay, and when times are fairly nice and copacetic, and there's not a lot of stress in the ER, there's actually a whole bunch of BIP. Okay, enough BIP to bind to IRE. So when the endoplasmic reticulum is keeping up with protein folding and managing the proteins coming into the ER and moving them on and doing that extremely well, there's an excess amount of BIP. So BIP actually then binds to PERC, it binds to IRE1, and it binds to ATF6. Okay? And the binding of PERC, of IRE to these three proteins tells these three proteins that things are fine and copacetic. Okay. But as unfolded proteins begin to accumulate in the cell, or in the ER, okay, then BIP leaves IRE, okay, because BIP is off trying to fold other proteins. Okay, you can draw a whole bunch of BIP protein complexes. Okay, so BIP is going to release from the IRE and bind to some exposed hydrophobic set of amino acids on a misfolded protein. Okay, so that leaves two hydrophobic regions in IRE that have to be buried. So one way to bury those hydrophobic regions is to dimerize. Okay? So two IREs will dimerize through that hydrophobic region that used to be bound by BIP. And as we're going to see in the next unit also, dimerization is an important signaling event. It's a real common feature. You'll get the hang of it. You can almost predict when dimerization is going to happen and what's going to happen. So we get a dimerization event. Okay, so BIP generally keeps the proteins from being dimerized, and the loss of BIP allows dimerization. And we see a conformational shift and IRE becomes phosphorylated. Okay? IRE phosphorylates each other. Okay? So these two domains on the cytoplasmic side are actually protein kinases. Okay? And the job of this protein kinase is only, I think as far as I know, it's only to phosphorylate each other. Okay, so this IRE phosphorylates this IRE, and vice versa. So we call that trans, okay, autophosphorylation. Okay, so you bring those two protein kinases into close proximity with one another, and one protein kinase phosphorylates the other. So that's the trans part. And the autophosphorylation is that IRE phosphorylates IRE. We're going to see the same thing happening when we activate the insulin receptor.